Mipomersen, targeting hypercholesterolemia through ASO magic.
Good morning, doctor.
Good morning, patient. What brings you here today?
I've noticed some unusual symptoms such as yellowness on my eyes and yellow lumps on my body.
These are likely to be xanthomas, which are a buildup of lipid droplets under the surface of the skin. Have you had any other symptoms heart related?
I've had some heart problems since childhood, and my family has a history of heart disease.
We got to get your cholesterol levels checked. So I got your report here, and your cholesterol levels are very high, well over 145 milligrams per deciliter, and normal levels are around 100. This is really unusual, and we need to do some genetic testing because I believe with your symptoms that you have homozygous familial hypercholesterolemia.
What's that, doc? And is there any treatment for that?
Yes, Mipomersen.
So what is homozygous familial hypercholesterolemia, also known as HoHF? HoHF is a rare genetically inherited disease, characterized by a mutation in both alleles of the ApoB100 gene. It is usually inherited in an autosomal dominant fashion and also displays some codominancy, with homozygotes expressing higher ApoB100 levels than heterozygotes. As someone with the homozygous version of this disease, it's likely that both your parents had some issues with heart disease. This disease is caused by LDL-C specifically, also known as low-density lipoprotein cholesterol. LDL-C is considered bad cholesterol because it brings cholesterol to tissues and it forms plaque deposits on the artery walls that can burst, causing the formation of blood clots, which clog and reduce blood flow through arteries. When blood flow is limited, increased risk of heart disease becomes a concern. Genetic conditions like HoHF can cause this cholesterol buildup. So where does ApoB100 come into this? LDL-C is a lipoprotein. This means that fatty cholesterol components must be bound to ApoB100 protein, forming an intermediate called very low density lipoprotein, or VLDL, in order to leave the liver, where it's metabolized in the bloodstream to LDL-C. So what can you do if you're diagnosed with this disease? After scientists understood the genetic basis of HoHF, they decided to target the ApoB100 gene, which is transcribed to ApoB100 mRNA. Under normal conditions, the mRNA codes for the formation of this crucial ApoB100 protein component of LDL-C. The discovery of antisense oligonucleotides, also known as ASOs, played an important role. ASOs are an artificially constructed sequence of nucleotides designed to complement a piece of mRNA. After identifying the sequence of ApoB100 to be targeted, the scientists at Ionis designed Mipomersen, a synthetic antisense oligonucleotide that targets the ApoB100 mRNA. Oligonucleotide binding leaves the mRNA inaccessible to the ribosome, blocking translational machinery. It also creates a binding site for RNase H, an enzyme present in the cell that degrades the mRNA, inhibiting production of the ApoB100 and LDL-C as a whole, ultimately reducing the levels of cholesterol in the liver. While mipomersen inhibits the production of LDL-C by stopping protein production, these fatty intermediates build up in the liver and have no way to leave, causing hepatotoxic effects. Mipomersen is currently used in conjunction with statin therapies, which are designed to lower lipid levels, but are known to have adverse effects. Mipomersen can be improved by finding novel methods to control the levels of these fatty intermediates. In 2013, Mipomersen began being marketed under the name Canamro, as it was approved by the FDA as a treatment of HoHF in patients who require additional lowering of LDL-C levels in combination with other lipid-lowering medications, such as statins. Mipomersen can be considered an orphan drug, meaning it is only used to treat rare medical conditions like HoHF. Mipomersen was tested for effectiveness in clinical trials before entering the market. In phase 1, mipomersen demonstrated dose-dependent reductions in ApoB100 and LDL-C, with the most significant effects at 200 milligrams. Adverse effects were mostly mild injection site reactions. However, phase 2 and 3 trials revealed additional adverse effects, including irritation at the injection site, flu-like symptoms, and hepatotoxicity, leading to elevated liver enzymes. Due to these concerns, the FDA flagged mipomersen for potential hepatotoxicity, indicating a risk of liver-related side effects associated with its use. Currently, ASOs find limited application, primarily delivered by intravenous or subcutaneous injections due to their inherent instability and susceptibility to degradation. The origins of mipomersen's injection site reactions remain elusive, but ongoing research aims to enhance drug formulation, potentially offering a solution. Furthermore, an alternative under consideration involves the incorporation of ASOs into oral pill formulations. However, this approach requires resource-intensive research to design chemical modifications capable of safeguarding against degradation. In conclusion, the journey from genetic understanding to ASO-based therapy exemplifies the strides made in targeting HoHF. While mipomersen's approval marked a significant milestone, the pursuit of refining its efficacy and delivery methods remains